Hello. It's really a tremendous pleasure to be here at Biodesign. As Dan said, I watch or have been paying attention to all of your projects over the past year as we promote them on issues. And then it's just an enormous pleasure to get to participate and to watch all of the work that you've been doing and all of the thinking and I'm intrigued. I feel happy when I see biodesign projects, not because they're happy projects, but because we're taking some active role in thinking about our future and thinking about the world and the joining of technology and art that we are doing, and instead of just like letting it fall on us. And so let me turn to Kathy High, who has been thinking about how do we keep the world from falling on us for many, many years. Her art has been exploring all sorts of fascinating things. And it's kind of driven by a feminist queer sensibility, an interest in a dialogue between art and technology, as well as community engagement, very deep community engagement. And we'll sort of get to that at the end. She's also, well, let me just back up. So when microbiologists talk, or when biologists talk about the deep past, they talk about a LUCA, last universal common ancestor. 4 billion years ago, something came to life. There's a little twist of DNA and something more. And then it began to reproduce, and suddenly there was life, and we all have some like commonality back to that space. Kathy has a universal common ancestor quality in the bioart space. There are a lot of people who've worked closely with her, been inspired by her, bounced off of her ideas. Among these people are Pia Interlandi, Heather Dewey-Hagborg, Ellie Irons. She introduced me to someone who does ant music, named Lisa Schoenberg, which I turn on the ant music when I need some ant music. Kathy, very nice to talk to you. I was thinking, let's just get started with rats. How did you get interested in rats?
Well, thank you so much, Lisa. And it's such a pleasure to be here, as you said. And thanks to the Biodesign Challenge for inviting me. And also, likewise, I've been part of the Biodesign Challenge since the beginning, watching Dan create this amazing space here for students. And also now this team he has with him is just stellar. So I'm really proud to be here, and it's really a pleasure to talk to you. I love your book, Underbug, by the way, just to promote that. So going back to rats. So yeah, I mean, a lot of my work comes from, looking at my own body and thinking about what is going on with my own diseases and things like that. I think all of us have this sort of firsthand information very intimately with what we are dealing with. And for myself, I became interested in rats, partly because I started to get really interested in what research was taking place on their bodies. They're like these scientific models and products that are used within research. And I got really intrigued with how many of them are used and how come I didn't know about this. I didn't know anything about this until I started sort of looking into the pipeline, let's put it that way.
Okay.
Because a lot of this research is kept hidden, invisible. So that's partly what got me interested in rats.
And then where did it take you? I mean, when I look at your page, there's all kinds of rat work and you had maybe 15 years of evolving relationships with rats, and you have Tamagotchi rats and you have white chocolate rats, but you also have real rats. Can you tell me a little bit about how, so let's just back up for a second. How many lab rats are there right now?
Nobody knows.
Okay, I think you mentioned
totally
8 million.
Yeah, there's guesstimates at something like that, but really they're not really counted because there are many different universities, research centers, etc., that use rats, mice, and birds as subjects among other animals, and we don't really know how many are used.
So basically, every molecule that's getting tested for any use, it ends up being given to a rat, essentially, to see how the rat reacts to it, or a collection of rats, hopefully, reacts to it. And then that is used as kind of a proxy for the human experience of that molecule.
Yeah,
So
I think that's a good definition of it.
and
Yep,
it's running
exactly.
at the rate of maybe 80 million human rat futures a year. Totally crazy. Yeah.
Yeah.
So tell us about your rats.
So, well, I just remembered the other day that actually some of the first rats I'd ever encountered were some students of mine had dyed their rats, their albino rats, pink for a shoot, and they were going to throw them out or something. And I was like, no, wait, let me take these rats. So this was a number of years ago. So I actually had some experience with them. But the rats that I ended up getting for this project called Embracing Animal were rats that were used in this kind of research that I was talking about. So they were used for autoimmune diseases and developed to be sick for treating rheumatoid arthritis, inflammatory bowel diseases, et cetera. And I was able to just kind of, at the time, slightly under the radar, order some and start to work with them. And I had these theories, and they're totally, not scientific, but I was interested in looking at sort of it from a perspective of a humanist. What happens to these rats if we take them out of the lab and start treating them alternatively? That's what I was doing with myself. I wasn't having a lot of success with the medical system in treating things like my Crohn's disease that I have. And I kind of started things like acupuncture long time ago. And that started to work. And even my doctors were like, why is this working? And I'm like, I can't tell you, but it is. So I thought maybe we could start working with the rats that way and doing some of these experiments. It did kind of work. It wasn't a controlled situation. I don't have any real statistics, but basically the rats basically thrived. And it was an amazing situation to watch them, become individuals.
So your goal was twofold. One was to make the sick rats healthy, and the other one was to experience the rats as individuals.
Yeah, I don't think that, I don't really think they could ever become healthy because they were bred to be sick. They could become healthier. So that was one goal. And to give them the opportunity to explore spaces and foods and each other in ways that lab conditions don't allow. And these animals can't really exist just in the wild. We couldn't really release them because they do have these diseases. So they had to be cared for, and we did care for them. And when I say we, this is in a situation where the rats were either with me at my house or in in a gallery or a museum, and they were cared for by the curatorial team, by veterinarians, et cetera, and myself. So it was a labor of love by a lot of people to try and build these large spaces for them, enrich their lives, and make them, just live their lives differently. That was one part of the goal of the project. The other part of the goal of the project was to try and translate some of this work that the rats were doing for the public. So as you said, this work started a long time ago, and I had the rats in 2004 to 2006. Between that time, they were exhibited twice, but one of the exhibitions was at the Mass MoCA here in Western Massachusetts, which is a museum that puts on long-term exhibitions. So this was an exhibition about animals and human-animal relationships, and it was up for 10 months. So they estimated 10,000 people come through a month. So that's a lot of people who get to see this work. And they were really great at training their docents to be able to talk about transgenic animals. So the rats had human DNA, so they could carry the kinds of immune diseases, autoimmune diseases, like my own. And so they were a little bit human. This is why I was attracted to them. They were a little bit like us, or part of us. But in 2004, transgenics wasn't understood in the same way it's understood now. It's more ubiquitous, it's more out in the public, and I think that it's easier for people to understand that term. At that point, it wasn't quite as new, as well versed publicly. So the museum did a great job with their docents and the people who introduced the work to actually bring that term to the forefront and make people understand this kind of genetic work that we're doing with our research subjects. And that now extends to humans too, of course. But, you know, we do that a lot with animals. I'll just say that.
I want to know a little bit about your relationship with the rats. I mean, there's a video on your site, and there's a little rat comes up to the edge of a cage and squeezes. I can't do it because I don't have a rat nose, but the rat's nose squeezes over the wire on the cage and really seems to feel it. And you can even see the little nose sort of go down. And you really get this sense that the rat is doing something and it knows what it's doing and it has feelings and it's thinking, well, who knows what the rat's thinking? I won't impose any thoughts on the rat, but the rat is exploring that environment very intentionally. And then you think, oh, all these rats in all these cages. And I guess my question is, what is the role of empathy in this work that's on transgenic animals and rats and humans and technology? What is it? What do you think the role of empathy is?
I love that question because I think empathy is one of the important things that we need to understand when we think about what I typically call as trash animals. And by trash animals, I mean animals that are really, you know, ones that we don't really care if they live or not. So rats, pigs, coyotes, you know, these kinds of animals would be put into this term, under this term. And I think that then we can even extend that to things like microbes and other cellular beings or viruses, and to develop a kind of care or concern that goes beyond the human, I think is really incredibly important to be able to empathize with the world around us. That includes people, of course, and all of the kind of disparities that we know are completely existing around us right now. But to change the system and to structurally change the system, I think we have to have other kinds of ways of thinking. And these other ways of thinking involve these kinds of interdisciplinary practices and looking at art, science, and humanities as part of a whole. So to get back to your question about that rat, that was Echo. And she was really great at sort of trying to get my attention when she wanted to we had this special little blueberry treat that she loved. And she knew exactly how to get me going and get her that treat. And she was incredibly smart. And when I first got rats, I didn't like rats at all. I mean, I was not a fan. I didn't know what to do with them. By the end of this period living with them, I had basically fallen in love with each one differently because each one of them was different. Echo, Star, Matilda, Tara, and Flowers were the 5 that I had had. So they were amazing as a team, and they taught me more than I ever taught them.
I think that's really interesting. I even saw that you did like sort of funerals for them, special funeral things. I wanted to talk a little bit about the you mentioned that empathy is not enough, and you need to have this interdisciplinary rubric to explore these things and to really work on how to create structural change. And a lot of that really comes from the social critique, which is so obvious when you're a judge or involved with biodesign, is like, what is social critique? Where do we do it? Why do we do it? What is our goal? There's good kinds of social critique and bad kinds of social critique, and there's so many interesting questions in it. But it struck me this morning, I was going to I have asthma, I get an injection every 2 weeks. And it struck me that we talk about biodesign challenge with the metaphors and the proxies for humans and all of these things as though it's a futuristic project. But when you think about the rats, it is a project we have been involved with for 50 or 60 years. We live because we have this intense relationship with rats. And some of our biodesign, this is very much a biodesign project where the humans and the rats don't quite collaborate. It's a very unequal relationship, but the passing of the molecules back and forth is a big deal. And yet at the same time, in my job, I happen to be editing a piece about how rats don't actually translate to humans. I mean, we use them as a metaphor, but they don't express genes in the same ways that we do. So it's it's an interesting thing that this has been going on for a long time. And the biodesign it's not necessarily futurist. It's just that it's been very much uncritiqued. It's very new to social critique. And so with that, if you want to add, do you want to add something, or I was going to move on to microbes?
No, I think you're right. I mean, I think that the one thing that's interesting about this kind of speculation into the future is that it allows us to create a space where you can begin to ask these questions. You know, whether it's through Afrofuturism, whether it's through science fiction. I think that you can create a space to critique things at a slight distance, and it gives people, affords the audience can not feel quite as implicated sometimes, and that's both bad and good. So I think that we have to think about what those situations do. At the same time, I think this kind of social critique within work is incredibly important, and one of the key things that we need to keep in mind as we go forward, especially art that provokes and or asks questions. So that's something that's important to me. Yeah.
Yeah. Let's talk a little bit about your work with microbiota. How did you get involved with that? Was that another thing where it was experiential and then it became kind of the social critique?
Yeah. So it's so funny you're picking up these things. I'm sure I sound like a weirdo to everybody, but good, great, thank you. But I really do, you know, that work came about because I had been working with cells, with immune cells, white blood cells. And then right about at the time I was doing that, there was all this research going on around the gut microbiome. And of course, having Crohn's disease and other things and all of this, I was fascinated by this and got fascinated also with fecal microbial transplants. So that is a process, for those who don't know, of taking poop from a healthy person and actually implanting it in a sick person so that it repopulates your gut with good bacteria and other things, hopefully. Doesn't always work, but in many cases and with some diseases, it works like a charm. So it's amazing. And I had never heard of that kind of a cure. I'd been through so many different, the technologies around how to treat autoimmune diseases are really weird, as are the treatments. So to think about this, which is like a paradigm shift about how we look at medicine, because we're actually treating our bodies with our own bodily material. And I know that that's been practiced in a lot of different medicine, but in this case, it's very firsthand, literally. So I found that fascinating. And then I got further interested in what's in that gut microbiome. What are the bacteria doing? How do they live together? And all of this kind of stuff. And it's still super new, even though the research has been going on for a long time, in decades. We're still at the very tip of the iceberg, which is really exciting.
What are there uncomfortable questions you're sort of pursuing with your art on the microbiome?
I guess one of them would be what happens with people's from different people's situations and disease types. Sometimes your gut is going to be stripped of a lot of bacteria, particular kinds of bacteria. If you've been treated with antibiotics over long periods of time, repeatedly, that can happen too, not just from disease. Right. So what happens when you look at the profiles of those guts? It makes me really uncomfortable to think, like, how can we help to repopulate these people's guts? Probiotics are great, but I'm not sure that they're long lasting, so or have any effect. So, how are we going to do this? And who's going to curate these bacteria that are so-called going to go back into us? And who's going to make those decisions? So I think that it has to be a collective decision, but that means changing this whole system, as Janina was talking about in her talk before, of trying to, what was the term she used, decolonize the language. It's the same kind of principle. We need to all be involved in these kinds of practices like a citizen science where, or community science, better, because not everybody's a citizen, where people can start being involved in these decisions.
And that is actually one of the interesting things about the fecal trades, is that they were done by sort of citizen scientists on their own, and it became like this sort of social movement. I thought it was really interesting. I think also, you know, here again, just like with your work on rats, you're making something that's invisible visible. I mean, I love the fact that your uncomfortable question was not putting individual poos in honey in special glass jars, which you actually did in a museum.
Yeah, okay, that's true.
But you know, one of the things I find really interesting about the evolving sense of the microbiome is how it's kind of changing what it means to be human. You know, at one point I was interviewing a microbial ecologist, and we were drinking coffee, and he was talking about how, you know, we think as humans that that we want coffee or we want potato chips, but actually it could just be your gut saying like, hey, bring us coffee or bring us potato chips. And there's sort of another thing, Janina was talking about us having 25,000 genes, but we also have about 9 million genes, microbial genes inside us and outside us, and there's viruses attached to them and related to them. And so we end up being about, you know, 400 times bigger than we think we are, which is wild. I mean, there's sort of just as the rats are, and we're thinking about a new kind of life, this transgenic business, we're also becoming kind of diffuse. We're like a cloud of stuff, and we don't quite know ourselves.
Totally. And that also disrupts this whole, you know, kind of European thinking, you know, philosophy of the self, and that we're on top of the heap of everything, because we're not. We're made up of everything that's around us. We are a part of everything. And I think, it really needs to be rethought and thought through in terms of how we look at our bodies, how we then look at everything around us, because it's all leaking in and out all the time. We're a part of all of it, and they're a part of us.
It's all leaking in and out. So this is going to require sort of a new kind of social critique in which the individual human is not at the center of it. I mean, along with the decolonizing of the sort of stratification, imaginary stratifications or genetic classifications, we're also going to have to start kind of redefining what the unit is.
Right. Yeah. Like a kind of queer ecology. You know, like, what is it exactly? And queer in the sense of, you know, the way we are using it today, but also the way it also had traditionally been used as something that's different, you know, something that is a new look at this whole way we think about ecology. So I think there's a lot of opportunity there for work to rethink things.
One of the things I really love is that you're doing so much work with sort of community outreach, and how do individuals become, I mean, to the extent that we are individuals, become, you know, involved in this process of science and art and iteration and social critique and the designing of this ongoing relationship that we have. And tell us about your what you're excited about working on right now.
Well, I'm super excited because this very weekend we are opening a community science lab in North Troy, which is where I teach at Rensselaer Polytechnic Institute. And so this is separate from the university, and it's a community project and a nonprofit with a group who are really wonderful, an amazing, huge group of people called the Sanctuary for Independent Media. We started as media activists and progressive media makers. And over time, we realized that we had to sort of also include this branch of ecology, because it was becoming imperative that that become work that we do as well. So we have a low power radio station and produce podcasts, and it's really, really amazing. We have a weekly show, but we also are opening an environmental education center called Nature Lab. And that's what's happening this weekend. And we've spent, you know, the past few years, and even during the pandemic, time grant writing, raising funds to open up this space, which is actually a little laboratory, like a biosafety level 1 laboratory in this incredibly, it's like one of the hardest hit areas in New York State, in this community that we're in. And it's a project that has the laboratory on the first floor, and then a group called the People's Health Sanctuary. We're all working on, and this is what's going to go into the second floor, which is still being constructed. And this is a group that looks at healing and alternative healing and also trauma, particularly. So I think that those two things coupled together, the science that we'll be practicing and the kind of skill sets that we'll be hopefully being able to review in curriculum and workshops, and then this look at what trauma has been caused to all of us through the pandemic, through the deaths of everybody who's passed in the pandemic, and so on, the kind of social justice and environmental justice inequities. And the projects are looking at, how to think through these very deep concerns that I think we all share, and train other generations to come aboard and be able to enter, if they want to enter the corporate system, if they want to enter academia, this is another way to gain training outside of, the school systems. So we're hoping this will work. We've been doing it for a number of years outside of the lab in just little workshops, but now we actually have this space, and it's an incredibly beautiful project that makes me really happy that we're doing it. So go to mediasanctuary.org and you'll see that. You'll see all of that.
I think this evolution that you're talking about, from art to art with the technology to art and communities, and you've got the communities of microbes and you've got the communities of humans. It's a really interesting evolution, and it helps us think about moving well beyond empathy into some sort of action.
Yeah, I think that's a great way to put it.
Kathy, thank you so much. It's been a huge pleasure to talk to you, and I hope that everyone can go to Kathy's website and see how stunning some of this art is, and watch Echo the rat put her little nose on the wire.
Lisa, thank you too. Really a pleasure.